third-party content has been copied and communicated pursuant to Part 5A or 5B of the Copyright Act, unless indicated otherwise.
In this set of learning objectives, we're going to cover functional brain anatomy, with a particular focus on the forebrain cortex. This is kind of dry material, but it is fascinating, particularly in the clinical context. What I'm proposing to do is to just, in these learning objectives, I'll go over the bits that you need to learn and know for the exam in particular. In the seminar, I'll use it as a starting point to go into some details and some clinical examples that are of interest. The central nervous system can be divided into three parts: the hindbrain, the midbrain, and the forebrain. Overall, in vertebrates, these three parts are generally of similar size. But in humans, the forebrain is most pronounced, with the mid and hindbrains representing a disproportionately small area of brain tissue. But this doesn't mean that the mid and hindbrains are not important, even to us. These regions are considered old, in evolutionary terms, in the sense that they were developed and specialized first. But they also serve very important functions that are important and essential for survival to this day. So let's go over some of the key structures and their functions in these three parts of the brain. Before we begin, just a note that not all of the structures that I have labelled in these diagrams actually belong to the areas we'll be talking about. Some of these structures are just labelled and included just as a reference. Okay, let's start by looking at the brain on the left. It's a photo of the brain, and on the right is a diagram of the mid and hind brains with the forebrain removed. So it's always good to compare photos with actual schematics. So on the left is showing a left-pointing brain that's been split in half. So you're seeing the inner side, the inner face of the brain. You can see the general area occupied by the midbrain. It's actually very, very tiny. It's just that tiny little area near the center of the brain. The two main components of the midbrain, and they're labelled quite nicely on the right diagram, are the superior and inferior colliculi. Superior because they're above, inferior because they're below. Both of the colliculi, both pairs of them, are considered primitive sensory areas. The superior colliculus is a primitive visual area. The inferior colliculus is a primitive auditory area. By that I mean it's not a very conscious part of the brain. It allows us to engage in reflexive behaviours, automatic behaviours. So if something buzzes past you, it catches your eye, you orient towards it. Or if you hear a loud sound to your right, you instinctively turn towards the sound. These very automatic, not quite conscious responses that we refer to as orienting responses, they're subserved by the superior and inferior colliculi. In animals like sheep, you see that the superior and inferior colliculi, particularly the superior visual ones, are actually very large. The hindbrain also includes the pons and the medulla. Both of these areas show up, and you can see on the left there, show up as very white rather than grey. They're white because they're made up primarily of myelin, which is a white fatty substance that insulates axons. So the pons and medulla contain primarily axons, or cellular pathways. Another area shown on the left, which is actually part of the forebrain, but I'm only mentioning it because it's so distinctive, is the corpus callosum. It's made up of axons as well. You can see it's very white, and it serves, it's colloquially referred to as the superhighway between the two hemispheres. And it's basically the axons that interconnect neurons between the left and the right hemispheres of the brain. This white matter contrasts with most areas of the brain, which are actually grey because they contain cell bodies. The medulla is particularly important and critical for survival. It controls reflexive breathing and heartbeat. Damage to the medulla can result in things like coma, even death. These structures of the mid and hind brain are more easily discerned on the diagram on the right, again because the forebrain's been stripped away. And you're looking at that diagram on the right, you're looking at the brain from behind and from the left side to give you a bit of a 3D perspective. The most complex component of the hindbrain, and it's visible on the photo on the left, is the cerebellum. The cerebellum is a very old region of the brain, but it's actually very, very sophisticated. It has many roles, but it's primarily concerned with the coordination of movements. If you have damage to the cerebellum, cerebellar deficits, it results in clumsy, imprecise movements, slurring of speech, things of that sort. You can see that it's very densely packed tissue as well. They're referred to as the vermis of the brain, the little worm-type shapes in the brain. In fact, it's amazing when you think about it that the number of neurons in the cerebellum is equal to the number of neurons in the grey areas of the brain, in the cortex of the brain. And you can think of it very much as a second brain, a second slightly smaller brain. When your primary brain wants to do something, it sends signals down the spinal cord to the rest of your body. But it also sends signals to the cerebellum. And when information from your body goes back to your brain, that information often also goes to the cerebellum. The cerebellum is in the position of being able to compare what your brain wants to do with what it actually achieves. And that ability to compare what you want to do and what you actually did allows the cerebellum to monitor and modify and regulate things like behaviours. We'll end with a review of the cerebral cortex. That's the surface of the forebrain, those 6 layers of cells I was referring to before. It's obviously grey because they're predominantly brain cells here rather than axons. And it's convoluted because these 6 layers of cells are trying to fit into the confines of a skull. So it's like a piece of paper that's been scrunched up. The grooves are referred to as sulci. The bumps are referred to as gyri. The 2 main sulci are the best landmarks you can use to identify the boundaries between what are called the lobes of the brain. The central sulcus divides the frontal from the parietal lobes. The lateral or sylvian sulcus divides the parietal from the temporal lobes. Let's go through the functions of the lobes, starting from the back and moving towards the front. The occipital lobe is the most specialized and it's the easiest to define. It's the primary site for visual processing. So basic visual processing occurs in the occipital lobe, and it is where the primary visual cortex resides. The temporal lobe houses the auditory cortex. So yes, it is also specialized for hearing, but it also is involved in other functions. In fact, it's also involved in vision. The whole bottom part of the temporal lobe, what's called the inferior temporal, is important for higher level visual processes, such as your ability to process faces or body parts, hands, that sort of thing. The temporal lobe is very important to that. The parietal lobe gets information about the body, the movement of the body, the location of body parts, of your own body that is. And it combines that information with visual information about the world and what it can see itself doing, that is, by looking at your own hands. So the parietal lobe is very much an associative area. It integrates information from touch, from hearing in particular. It helps you to decide, for example, whether an object is within reach, and if it is within reach, how should it be best grasped and manipulated. The frontal lobes are important for movement. It's where the primary motor cortex resides. It lets you make very fine, precise movements, such as the movements that control the pen that's in your hand. But the frontal lobe is also a very high-level cognitive center. It's important for things like memory, for planning, for high-level decision-making. It is often the part of the brain that we attribute your personality to. It's the most recently developed part of your nervous system in the evolutionary sense. And it's also the most associative. So it's actually the hardest to define. It's involved in a whole lot of things in a very high level sense. Finally, I show how both the motor and somatosensory cortices are topographically mapped, corresponding to particular body parts. In these figures, the location of the body part corresponds to the location on the cortex of the neurons that subserve that body part. And the size of the body part represents the actual number of neurons that are involved in relation to that body part. So you can see that, in terms of touch, the fingertips and the lips are particularly important. And you can see that in terms of movement, the hands and the fingers are very important. Now to the forebrain. And again, let's return to the photograph of the brain that's been split in half. It's a left-pointing brain, split in half, and we're seeing the inner surface of the right half of the brain. The forebrain is generally more interesting to us as psychologists because of its role in things like higher level sensory perceptual processing, its role in emotions, cognitions, planning, and fine motor production. The surface of the forebrain is actually very interesting. The surface, the bit you can actually see there, those grey convolutions, they're made up of 6 layers of cortex. But let's begin, not by talking about those surface areas, let's begin by digging deeper into what's referred to as the subcortical areas of the forebrain. As in the previous slide, let's compare the left photo alongside a diagram. Diagrams are convenient, particularly with subcortical areas, because these subcortical areas don't really show up well anatomically. They're not anatomically distinct, with the exception of the hippocampus, really. They're not that, and the cingulate. They're not that anatomically distinct, so it's good to have a bit of a colour schematic of it. In the diagram on the right, you're seeing the brain front on, so it's like the person's looking at you. So again, that gives you a different perspective in terms of the view as well. The structures labelled generally are considered to form part of the limbic system. And I say generally because there's actually some disagreement as to which ones do and which ones don't exactly form part of the limbic system. In fact, the hypothalamus and thalamus is sometimes not included as part of the limbic system. So these structures of the limbic system, what are they about? What's so important about them? Well, they're relevant to memory, they're relevant to emotions, particularly emotions like fear, but also aggression. They're relevant to sex. There's some evidence that some of these structures are relevant to sexual orientation, even in humans, areas within the hypothalamus, for example. They're relevant to smell, to olfaction, in other words. The hippocampus, in particular, is a smell center. The amygdala, in particular, is an emotional center. But also space, location of objects. We have what are called spatial maps. Your ability to remember and navigate your way home. The hippocampus is critical for that. In fact, in your textbook, it gives some interesting examples where they looked at the size of hippocampi of taxi drivers in London. And in London, at least traditionally, taxi drivers, they didn't have sat-nav systems. They actually had to memorize road maps and then later obviously navigate using their memory of the road network and finding the quickest path from A to B. And in fact, in those taxi drivers, their hippocampi are larger than the average person. And when they're thinking about how to get from A to B, in a brain scan, for example, in an MRI, you can actually see the hippocampus lighting up as it becomes more active. So the limbic system, a collection of things like memory, emotion, space, and cognitive kind of spatial maps, and including things like sex, including possibly even sexual orientation, even in humans. The components of the basal ganglia are not very structurally or anatomically distinct. They're identified primarily on the basis of their function rather than their anatomy. They're important for things like the planning and initiation of movement. People with disorders of the basal ganglia exhibit signs of things like Parkinson's disease or Huntington's disease, where they don't move properly, or they have trouble initiating movements, or their movements are exaggerated or uncontrolled with tremors, for example, or they have to shuffle when they walk. So there's a whole cluster of movement disorders that are associated with the various problems that can occur with the basal ganglia. So it's about planning and initiation of basic coordinated movements. You'll notice that the thalamus gets a mention and is both labelled and included in diagrams like this, and including the limbic system on the previous slide. This is because the thalamus is actually involved functionally in many contexts, because it's an intermediary, a neural hub, so to speak. So many pathways go through the thalamus, so you can classify it anatomically and functionally as belonging to quite a few systems and subsystems. From the previous couple of slides, you'd be forgiven for thinking that the brain is a disparate, disjointed, incoherent mess. But obviously, functionally and consciously, we are quite the opposite. We are extraordinarily coherent and integrated. If coherence and integration are the outcomes of all this disparate neural activity, why not make things easy by carrying out analyses on objects or whatever in the same part of the brain? Why have things like colour, motion, shape, location, touch, memory, knowledge, etc. about an object dealt with by such different parts of the brain? The answer is that the brain is a work in progress. It's the result of many renovations, many additions over the years. As our needs have changed, we've tended to simply tack on new functional modules to do the job, rather than attempting to overhaul or improve existing modules. The result is a great deal of redundancy, with similar tasks being carried out in different parts of the brain in different ways. For example, we've got several perceptual systems, we have several movement control systems, we have several memory cognition type systems. This raises the question, of course, how does it all then come together? How does the brain know that the disparate groups of neurons that are firing away are telling it about the one same aspect, different aspects of the same object? How does the brain know that these are just different stories about the one thing, rather than different stories about different objects? This is a bit of a paradox, a bit of a problem, and it's referred to as the binding problem. We don't have any solid answers, but one promising idea is that the different neurons involved in the same task are temporally synchronized. This is regardless of how much they're active. They're in temporal synchrony with each other. Think about an analogy, the different players in an orchestra, the clarinetist, the flautist, the piano player, the saxophonist, or whatever they happen to be. They're all playing different notes. They all sound very different, but they're maintaining the same tempo. And by maintaining the same tempo, they're telling you that they are all part of the one piece of music. They're all part of the same orchestra. Same thing with neurons, presumably.